Okay, I know you're so excited that you're just gonna jump right on ahead and get it to video too because you can't stand it. So let's just move on along. Maybe. There we go. Okay. Now today we're going to talk about DNA replication. This is making an exact copy. We're replicating something. So we're making an exact copy of DNA. Extremely important because we have to make new cells all the time. And so we have to replicate. Now, this process occurs in what's called the S phase of the cell cycle. We haven't studied the cell cycle yet. That's coming up. But just know that S is for synthesis. That means to make. So we're making DNA during this phase. All right? Now, this this process of making the DNA, this is what's called complementary base pairing. So we're making a complement. We're starting off with one parent and we're going to split that in half. We're going to open that up and then we're always going to keep one of the original strands as we make the new strands. This process is called semi-conservative, and it's this model, the semi-conservative model that was proposed in 1958 by Matthew Meselson and Franklin Stahl. We just hear him referred to as Meselson-Stahl, but so it's called the semi-conservative because we're still maintaining the original. You're going to see this is real important when we're talking about proofreading errors. All right. Now, another thing that I need to point out about Meselson-Stahl, this semi-conservative model. First of all, we know that A pairs with T and G pairs with C. The other thing that we want to look at is that this is occurring, this replication work is being done in opposite directions at both times. So it's going to go in one direction this way and the other direction in this way. And it's occurring at the same time. Now in humans, this is real important. It takes just a few hours to copy over 6 billion, that's with a B, 6 billion nucleotides in our cells, thanks to enzymes. So when we say that enzymes lower the amount of activation energy, we're not kidding. So over 6 billion nucleotides in just a few hours. Very important. All right, so let's look at the origin of replication. So whoa, moving on along. The origin of replication. Where does it begin? And these replication bubbles. All right, now this is the starting point. So there are specific nucleotide sequences that are encoded in the DNA, and their job is to start the process. That's their code. So there's different codes that will that will give different commands, and theirs is the start code. Now this is not going to, this shouldn't seem like an amazing discovery, but there is an enzyme called helicase. Now I want you to think about this. ACE, of course, enzyme, HeLa. What would that refer to? Think about it. We have a DNA double helix. So helicase unwinds the DNA double helix and it creates what's called a replication bubble. So what it does is it actually opens it up and provides spaces to do the actual building work of making the new complementary side. So it's going to open up and it's allowed. Now remember, there's still this side, semi-conservative. All right. So in this DNA, we have this elongation using DNA polymerase. So you have elongation of the new complementary side. So we're going to elongate this, and it's going to require the enzyme DNA polymerase. All right. DNA polymerase 3. So this enzyme actually performs the new addition of new nucleotides to the new DNA complementary side. So we're adding DNA nucleotides. So we're adding more T's and A's and G's and C's. We're adding those, and they also act as a proofreader to help prevent errors. So look at the name and you're going to see the function. Okay, remember poly means many. So we're going to have many units, many monomers. And in this case, the monomers are called nucleotides. And so we're making many of these nucleotides. We're gluing them in there. All right. So the enzymes work at about the rate of 500 nucleotides per second. That's fast. Now, the two sides of the double helix are said to be anti-parallel to each other. This means that the DNA is going to, the information runs in different directions. They're going to go, it's always red from the 5 prime to the 3 prime end. Okay, so it's always red in this direction. Now, if we look at here, the 5 carbon sugar, this can be DNA or RNA, but here's, we're looking here. Now this 5 carbon, so here we have this little sign after here is called prime. So 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. The 5 prime carbon of the sugar, whether it's deoxyribose or ribose sugar, has this phosphate attached to it. So the phosphate's attached to the 5 prime end. The 1 prime is attached to the nucleotide. So this one right here is going, to the nitrogenous base. So this one right here is attached to the adenine, thymine, guanine, or cytosine. And then this 3 prime right down here has an open bond. And it's here that becomes the connector site for the next nucleotide. All right, helicase. So this is our enzyme that does the unzipping. It's going to unwind this DNA. So this would be, so here, they're just showing this section right here. So this bubble's been created, it's opened up, and they just took one little section out of that so you can see what's happening here. All right, the lead strand is the top strand, and the lagging strand is here on the bottom. All right, so you have this single-stranded region. The binding protein keeps the two sides apart and stable. So, and then you have the leading strand of the, okay, let's go back over here. My son's dancing in the kitchen, so it's a little distracting right now. Gee, I wish I could show you in the video, but it would be frightening. All right, so the leading strand at the replication fork, remember there's 2 forks. There's one here, there's the other one here going in opposite directions. So this strand runs in a continuous 5 prime to 3 prime direction as it opens. It's leading the way in the process. In order to start adding nucleosides, we first need to attach an RNA primer, which would make sense. So here's our RNA primer coming in here. And it's using a primase. Well, that makes sense. A primase, an enzyme to prime it, and then go. So the lagging strand here, so this is the leading strand. The lagging strand, this side of the replication fork, has DNA not running in a 5 prime. It's not going in the 5 prime to 3 prime. So it's behind the other one. It's going to be lagging, just like the word, if you're lagging behind. This one's lagging behind. So this side of the fork has to wait for this long segment of DNA to be exposed before we start by adding a primer. So when a long segment's been opened by the helicase, making the bubble, the RNA primer, which is disposable, is going to attach, and then DNA polymerase 3 will work backwards by making what's called Okazaki fragments. So these fragments, these chunks, so instead of one at a time, you're making these Okazaki fragments. I love saying that word. Isn't that the coolest word? Okay, so now all of you should stop, should pause the video, and go to your parents and just say, oh my gosh, I'm exhausted. I have just been listening to all this Okazaki fragment stuff, and whew, it's so interesting. You know what? I just got to get back to it and then come back and watch the video and just watch how their jaws drop. Okay, oh, whatever. I'm weird. Okay, so these Okazaki fragments, they are stitched together using an enzyme ligase. Okay, so ligase is like glue. Okay, so it's gluing them together. So these come in, and then the ligase comes in there and glues them together. All right, so now you have all of this, the primer, everything's been made, And so now we have to proofread. So the function performed by the DNA polymerase, so this function is being performed as the proofreading. Oops, whoa, move down along. All right, so the mismatch repair is when the wrong nucleotide is added to the new sequence. So if there's a wrong nucleotide, then the DNA polymerase is going to reverse the spot, remove the wrong nucleotide. So it's going to back it up. It's going to remove this and then replace it with the correct one. It's kind of like if you misspell a word when you're typing, and then you just go back and you fix it. So for errors that are created, which are called mutations, after the DNA has been made, you have this nucleotide excision repair that's used to correct. So step one, you have this nuclease, which cuts around the faulty repairing. Step two is DNA polymerase, so it's replacing the missing nucleotides, and then of course the DNA ligase, which is gluing or sealing them back together. So errors in the proofreading can result in some forms of cancer. For instance, some individuals are genetically predisposed to skin cancer because they have a mutation in the gene that codes for this excision repair enzyme, and they can't fix which can't fix the damage caused by UV light on the skin cells. We're going to talk in more detail about cancer and what causes why we have a higher rate of it when we're exposed to different things or just over time. So that's coming up, so hang on to your hats. Woo!